Hey guys, we're back with chapter 4, single gene inheritance, but this time we're going to be looking at sections 2 and 3. So section 2 is single gene inheritance is rare, and section 3, following the inheritance of two genes. All right, so let's get going, shall we? So modes of inheritance are the rules that explain the patterns within an inheritance. So when we talk about something being dominant versus recessive, those are modes of inheritance. When we talk about autosomal, remember autosome means not on the sex chromosome, so autosomal dominant versus autosomal recessive, that's also talking about modes of inheritance. In our next chapter, we'll talk about even more modes of inheritance, such as codominance, incomplete dominance, epistasis. Later on, we'll even talk about sex-linked and how that's a mode of inheritance. So mode of inheritance are simply the types of inheritance. So the rules that we see when we notice a pattern. Mendelian trait is caused by a single gene. So one single gene will determine that trait. Now, in Mendelian genetics and Mendelian traits, we typically see a dominant versus recessive mode of inheritance, which allows it to recur in predictable patterns in every single generation. That's why we can essentially follow, why we can follow along with it, like with the Punnett square, and use that Punnett square to help us figure out what's the likelihood of the offspring. Because we know with Mendelian dominant and recessive inheritance that we should see certain traits coming up in certain probabilities. And let me put my camera back where it goes. All right. So autosomal dominant inheritance is one type of mode of inheritance that we're talking about in this chapter. So what this means is that you only need one dominant allele to show it. That doesn't mean you only have one allele. It just means that one of them will be a dominant allele. So whether you have two dominant alleles or one dominant and one recessive allele, regardless, you will see the dominant trait being expressed. Because of this, generally these don't skip generations, and just because with it being dominant, if you have the allele, you will show it. If you don't have any dominant alleles, that means you're recessive and you cannot pass on a dominant allele if you have none to pass on. So these really don't skip generations, and since it's autosomal, it will affect both sexes equally. So it affects males and females equally. We'll notice that in sex-linked traits, we don't have that same pattern. We notice that one sex is more affected than the other. In autosomal recessive inheritance, these can skip generations. And the reason for that is because if you are heterozygous, you have one dominant allele and one recessive allele, the dominant allele will be shown. The recessive allele is going to be hidden, but you can still pass it on to your children. So, for instance, my grandfather had blue eyes. My dad did not. My dad had brown eyes. I have blue eyes. So what happened is my dad was heterozygous. He got a brown eye allele from my grandma, from his mom, and the blue eye allele from his dad. So my dad was heterozygous. My dad gave me his recessive allele. And then I got a recessive allele from my mom, so I ended up being blue-eyed like my grandpa. So we saw that the blue eye trait skipped the generation. The allele was still there. There was still an allele present, but it was being hidden by the presence of the dominant allele. Since it's autosomal, once again, it can affect both sexes equally. So, for instance, blue eyes, you don't see males or females having a higher rate of blue eyes than the other. You can Males and females each have an equal opportunity to be blue-eyed. It doesn't affect them because it's not associated with the sex chromosome. Blood relatives that have children together will have a much higher risk of having children with rare recessive disorders. So what that means is, this is one of the reasons why incest is so bad, because let's say that within your family, there is a recessive allele for a horrible, horrible disorder, one that would kill the individual. It's fatal by the age of 5 years old. Now, no one really knew that your family had this recessive allele because it's so rare and not a whole lot of people outside the family have it. However, if two people within the family were to get together, let's just put it that way, incest, then they have a much higher chance of combining both of those rare recessive alleles together and thus allowing that disorder to be seen. All right, Punnett squares. I know we talked about this in our previous PowerPoint. We kind of talked a little bit about how to set them up. We're going to talk a little bit about why we use them. Now, the nice thing about Punnett squares is, like we said, these traits have predictable patterns of how they will be passed on to the next generation. The Punnett square allows us to visually show that pattern. So in order for us to figure out the predictability, the likelihood for different disorders to be seen, we can do a Punnett square and we can figure out the likelihood because of these predictable patterns. Let's see, so Mendelian traits apply anew to each child. Now, that's related to the Punnett square. Remember how the Punnett square has 4 squares? We are not saying that the parents will have 4 children and that is the exact genotype of each child. We're saying that we can do the Punnett square for each child. So 1 Punnett square for each child. So if you remember, when we had 2 heterozygous individuals, there was a 1-to-1 genotypic ratio. 1 out of 4 chance of having being homozygous dominant, 2 out of 4 chances of being heterozygous, and 1 out of 4 chance of being homozygous recessive. So that's 25%, 50%, 25%. We are not saying that that couple will have 4 children and 1 of them will be homozygous dominant, 2 of them will be heterozygous, and 1 of them will be homozygous recessive. What we are saying is that for each child, we can apply those ratios to it. So each child that is born has a 25% chance of being homozygous dominant, a 50% chance of being heterozygous, and a 25% chance of being homozygous recessive. So we're looking at that entire Punnett square, not the little mini squares in it. We're looking at the entire Punnett square and applying that to each child individually. At the bottom, I give you a couple more examples of single gene autosomal traits. So widow's peak is when you see that little dip down in the hairline versus a straight hairline. So I got a bit of a widow's peak myself right here. See how it dips down? And then hitchhiker's thumb versus the straighter thumb. Now you might remember that we said that single gene inheritance is rare. And that's mostly because we are not pea plants. It's very common in pea plants, which is why Mendel studied them. But humans, our modes of inheritance are all over the place. It's not just single gene. It's not just autosomal. It's not just dominant and recessive. We have codominant genes, we have incomplete dominant genes. We have multiple alleles, lethal allele combinations. We have multiple genes that will affect one trait. We have environmental effects that will affect one trait. So it's a lot more rare in humans. Now, does that mean we shouldn't study Mendelian genetics? No. It is great for teaching the basics, for introducing kids to the world of genetics. All right, but let's move on. Biochemical level. So why is it that dominant and recessive disorders function differently? A lot of it has to do with the proteins that are created as a result of a recessive versus dominant combination of alleles. Sorry about that. So typically, what we notice with recessive disorders is when you have two recessive alleles and it results in a disorder, there tends to be a loss of function for a protein or the loss of the production of that protein. So regardless, the protein is not there, or it's there, but it's not doing anything. Imagine you and your sibling. Now, you guys have this nice system. Your sibling washes the dishes. You dry and put them away. Now, you work together to do your job of cleaning up after dinner. If your sibling just stood there and did absolutely nothing, will the dishes get washed? No, they won't. What if your sibling's just not there at all? They're missing. Now it's just you, but you can only dry and put them away. Will the dishes get washed? No. A good example of this is albinism, or being albino. What occurs in those cases is usually either the pigment protein is not made at all, or it is made, but it's not put into the skin. So it's not actually deposited where it should be in order to work. So maybe it's there, but it can't get to where it needs to. It can't actually get to the skin cells to do their job. As opposed to a dominant disorder. Now, this is when you have at least one dominant allele. That's all you need to express a dominant disorder. What happens with this is the protein is made, but it's an abnormal protein. It either does a new function or it functions in a way that interferes with other proteins. So remember, with recessive disorder, the job was not getting done at all, either because the protein wasn't being made, so it's not present, or because it's being made, but it's just not doing anything. It's non-functional. Here with dominant disorders, the protein is being made, but it's doing the wrong job. So let's go back to the example of you and your sibling washing dishes. Sibling washing dishes, you drying and putting them away. So maybe they gain a function. So they're trying to sweep the floor and clean the windows while washing the dishes. That's a lot, isn't it? They're not going to be able to properly do their primary job if they're trying to do these other functions as well. Let's see. Or if they interfere. So what if, whenever your sibling washes the dishes, they throw them like a frisbee at you? Are you going to be able to catch them all, to dry them and put them away? No. So they're technically doing their job, but it is interfering with you doing your job properly. Some examples of this would include like Huntington's disease, how it affects the neurological function. The proteins are there and they're doing a job, but it's not the correct one. So it's just kind of showing how, once again, we're looking at the Punnett square aspect. Father might be a carrier for a recessive disorder. Mother's a carrier for a recessive disorder. So they're heterozygous. Neither one of them have the recessive disorder, but they carry the allele for it, so that they have a 25% chance of their child having that recessive disorder. All right. Let's talk about independent assortment. This is another one of Mendel's laws. Now, Mendel's law of independent assortment considers genes transmitted on different chromosomes. So what we're looking at now is what we call a dihybrid cross. So we're looking at two genes on completely different chromosomes. They're not ones that necessarily affect each other. So I could be looking at the eye color gene and the gene for the number of fingers I have. So I can look at that and figure out what's the likelihood of these two traits and the different variations of them being passed on together. Sorry. Now you're going to notice that we're not going to talk a whole lot about setting up the Punnett squares because it's the same rules. It's just bigger. This is a dihybrid cross at the bottom. Now remember when we were talking about the monohybrid crosses, we said with a heterozygous individual, there was a 1-to-1 genotypic ratio and a 3-to-1 phenotypic ratio. Well, in dihybrid crosses, when both parents are heterozygous for both traits, we tend to see a 9-3-3-1 ratio. So what that means is, because of this wide range of possibilities, it indicates that this one chromosome with gene A has no influence on how this chromosome with gene B gets passed on. Now, if you're wondering what the 9-3-3-1 refer to, we're saying that 9 out of 16 have, or there's a 9/16 chance, or if we're looking at the boxes, there are 16 boxes here. So 9 out of 16 show both traits as being dominant. 3 out of 16 show first trait being dominant, second trait being recessive. 3 out of 16, first trait being recessive, second trait being dominant, and 1 out of 16, both traits recessive. So what we're noticing is that different chromosomes do not affect each other. Now let's relate this back to meiosis. Remember, we said the law of segregation says how a homologous pair separates during meiosis so that each offspring only gets one copy. Well, what independent assortment is saying is that when all the homologous pairs separate, no homologous pair is going to affect the separation of another homologous pair. So for instance here, we have big R, big Y, little y. So we have two different chromosomes and we're looking at two different genes. One is on this chromosome, one is on this. This parent is heterozygous for both. So how can we separate them? Well, we can separate big R, big Y, so that the gametes only have a big R and a big Y. Or we can separate them with the reds, little r, little y, so the offspring only have the recessive alleles. Well, what if it just so happened that the chromosomes lined up differently during meiosis? Then we can have big R, little y, or we can have little r, big Y. So that is different combinations of how we can separate these along. And that's not even taking care or talking about crossing over and the fact that we have 23 pairs, not 2 pairs. So there's a lot of possibility. Again, kind of looking at this, they're showing you the red is the dominant versus red is dominant versus purple as our recessive. So we can maybe they get separated so the two recessives travel together. Maybe they move so that the dominant travel together. Here we have recessive and a dominant and recessive. So we have a possibility of one parent of the parent passing on a little r and a little y. They can also pass on a big R and a big Y, or they can pass on a little r and a big Y, or they can pass on the big R and the little y together. So we're looking at all the possible combinations of alleles that the parent can give. All right, so dihybrid crosses. In meiosis, random assortment of maternally from the mom and paternally from the dad derived chromosomes result in gametes that have different combinations of genes. So that's what we've been looking at before. Punnett squares and probability are used to predict the recurrence of more than one trait. So we can look at the occurrence of the purple flower with a tall plant versus purple flower with a short plant, or white flower with a tall plant, or white flower with a short plant. Here they also show that as well. Let's see if we have a couple other pictures though. All right, so in the parent generation, we have our true breeding individuals. We cross them, we get our F1 generation, first filial generation, and they are heterozygous for both traits. Now, if we want to cross them, then we have to first separate their alleles. And this is kind of like math distribution again. Now remember, they're two separate genes. So and during meiosis, we look at the Y genes, the Y alleles will separate so that each child or each gamete gets one Y allele. For the R gene, it'll separate so that each gamete gets one R allele. So that's what we're going to do. We're going to combine one Y with one R. So this big Y could separate into the same gamete as this big R, which is why you see that combination here. The little y can also separate into the same gamete as the big R. So little y, big R. The big Y may be able to separate into the same gamete with the little r. Big Y, little r. The little y may be able to separate into the same gamete as the little r, little y, little r. And since we're crossing two heterozygous parents, we're going to see the same combination on that side. Just like a Punnett square before, we're going to bring the letters down and bring them over. Big Y down, big Y over, big R down, big R over. Notice we're keeping the Y's together and the R's together because they're two completely separate genes. Big Y, big R, little r. Big Y, big R, little r. Big Y, big R, little r. Let's see. Big Y, big R, little r. Little y, big R, big R. Big Y, big R, little r. Little y, little r. Big R, little r. And so on and so on. Now, when we're done with this, we can take a look at this and we can figure out the possibilities. So if we want to look at the possibilities of the offspring being dominant for both traits, so being yellow and being round, we're looking for boxes that have at least one capital Y and at least one capital R. So yellow and round. 1, 2, 3, 4, 5, 6, 7, 8, 9. So there's a 9 out of 16 chance that the offspring will be yellow and round. How about green? So recessive for the first trait, but round, dominant for the second. 1, 2, 3. 3 out of 16 that the offspring will be recessive green, but dominant round. All right, how about dominant yellow, but recessive wrinkly? 1, 2, 3. 3 out of 16 chance of being yellow and wrinkly. And what about for being recessive for both traits, so being green and wrinkly? 1. 1 out of 16 chance that the offspring will be recessive for both traits. And we see the same thing here. I'm not going to go over everything again. But again, we can look at this. We can say, all right, 2 I's are dominant, and so is having the triangle head. So 1, 2, 3, 4, 5, 6, 7, 8, 9 out of 16 have triangle heads with 2 eyes. 1, 2, 3 have the pentagon face with 2 eyes. 1, 2, 3 have the triangle face with 1 eye, and 1 has a pentagon face with 1 eye. All right, so I hope that helped you to understand this material. If you have questions, hit me up in Google Classroom or email. I'll also have a Science in Pajama video on how to do dihybrid crosses, so maybe check that out. But in the meantime, you guys stay awesome, stay curious, and most importantly, stay safe and healthy. All right, you guys, take care. Bye-bye.